Good morning, students. In this video, we'll be talking about active transport. We have been talking about membrane transport since 2 or 3 videos. We talked about diffusion and osmosis, how they let the molecules move from high concentration to low concentration without any energy requirement. Now, in this video, we'll talk about active transport. So, active transport means it's a type of a membrane transport where the molecule is moving against its concentration gradient. Now, as the molecule is moving against its concentration gradient, that means it is moving from the low concentration to the higher concentration. It requires energy source. Energy source as a ATP, which stands for adenosine triphosphate, to move the molecules against the concentration gradient. So this is the cross-section view of the cell. The outer boundary is lipid bilayer membrane. Let's say this is the inside and this is the outside of the cell. Let's say, for example, in case of an animal cell, sodium concentration is less inside and sodium concentration is very high outside. So in this case, we want to transfer sodium outside the cell from the inside. So we are going against the concentration gradient. For achieving this task, we need energy. Unlike, you can see a potassium ions is high inside the cell and potassium concentration is low outside the cell. So if potassium wants to come inside against the concentration gradient, it requires energy. For instance, suppose the sugar glucose is more concentrated inside of a cell than outside. If the cell needs more sugar in to meet its metabolic needs, how can it get that sugar in it? Here the cell can't import glucose for free using diffusion, because the natural tendency of the glucose will be to diffuse out rather than flowing in. Instead, the cell must bring in more glucose molecules via active transport. In active transport, unlike passive transport, the cell spends energy in the form of ATP to move on substance against its concentration gradient. We have talked about how small molecules or ions might be able to go through a cell membrane in different ways, whether actively or passively, may be facilitated in some way. What we are going to talk now is how we can do this for larger things. We are going to focus on here is bulk transport. So this first example, you could say this cell with a cell membrane is engulfing this red things, which may be bacteria or something. And you can see the cellular membrane starts to wrap around this bacteria, then it fully wrapped around, and then that membrane that was wrapping around the bacteria pinches off, and now the bacteria is inside the cell and it's wrapped by this membrane. And this process where you are engulfing these large things, we call this phagocytosis. And the prefix phago comes from the Greek for to eat. So this is literally about cell eating. You could view this compartment that is holding this bacteria is going to transport to a lysosome so it can be digested in some way. We'd call this membrane bound compartment food vacuole. Now this scenario here is similar but different. Over here I have the cell made up of phospholipid bilayer. Let me make it very clear, this is inside the cell, this is outside the cell. And you can see here that instead of engulfing a large particle of bacteria, it's just engulfing some fluid. It starts to wrap around this section of fluid, wraps even more around this section of fluid, and then the membrane that was around it completely pinches off and goes into the cell. And this thing that has been pinched off and is now inside the cell, we call this a vesicle, which is just a general term for this membrane bound compartments inside of cells. And this process where the cell has essentially drunk a bunch of fluid, and the stuff that happens to be in the fluid, we call this pinocytosis. And pino comes from the Greek word to drink. So this is literally about cell drinking. This is phagocytosis, the cell is eating, and these are both special cases and the more general term of engulfing in this way, which is called endocytosis. So phagocytosis is a form of endocytosis, and pinocytosis is a form of endocytosis. Let's talk a little bit about exocytosis, which is how the cell is able to release large molecules that might be used by the rest of the body or the cells, or maybe it's going to be a part of the extracellular matrix. And to understand how this works, it's really the reverse of endocytosis. Suppose, for example, some proteins are produced in endoplasmic reticulum. So those proteins are then going to bud off in their own little vesicles, which then merge with the Golgi apparatus when they are further processed, and then eventually they are going to bud off the Golgi apparatus in new vesicles. And those vesicles are going to make their way the cell's outer membrane, the plasma membrane, and the membrane of the vesicles are going to merge with the membrane of the cell, and in doing so they are going to release their contents, and this is exocytosis.